Welcome to an in-depth look at molecular and cellular radiobiology. In this video, we'll break down complex concepts into easy-to-understand explanations. We'll cover the core principles of how radiation interacts with biological systems, from macromolecules to entire cells. First, let's explore the effects of in vitro irradiation on macromolecules, which are essential building blocks of life. One key effect is cross-linking, where radiation induces abnormal bonds between molecules, altering their function and structure. Another significant impact is main chain scission, where radiation breaks the backbone of macromolecules, leading to fragmentation and loss of integrity. Finally, point lesions involve subtle, localized damage that can change the molecular structure, potentially leading to mutations or malfunctions. Now, let's focus on the principal effects of radiation, specifically on DNA, the blueprint of life. Radiation can cause base damage and loss, altering the individual components of the genetic code and leading to incorrect information. More critically, it can cause single-strand breaks and double-strand breaks in the DNA helix, with double-strand breaks being particularly dangerous for cell survival. The radiolysis of water is a crucial chemical reaction in biological damage. When water molecules are irradiated, they break down into highly reactive free radicals. Next, let's examine the effects of in vivo irradiation, which refers to radiation exposure within a living organism. In vivo irradiation leads to both direct cellular damage, where radiation directly hits cellular components, and indirect cellular damage primarily through the free radicals from water radiolysis. At a larger scale, in vivo irradiation can trigger diverse tissue and organ responses, from inflammation and reduced function to severe damage and organ failure, depending on the dose and sensitivity. Now we turn to the principles of target theory, a fundamental concept in radiobiology. Target theory posits that critical cellular components, or targets, must be hit by radiation to produce a biological effect. The most sensitive target is often DNA. This theory includes single-hit and multi-hit models, which describe how many, quote, hits by radiation are required to inactivate a cell or target, influencing cell survival curves. Finally, let's explore how radiosensitivity varies across different stages of the cell cycle. Cells are most sensitive to radiation during the M phase, or mitosis, when chromosomes are condensed, and the G2 phase, where DNA repair mechanisms are not yet fully active. Conversely, cells are generally least sensitive during the S phase, due to active DNA repair, and the G1 phase, where cells have more time to repair damage before DNA synthesis. In summary, understanding molecular and cellular radiobiology is crucial for appreciating how radiation impacts biological systems, from the smallest molecules to the complex processes of cell division and tissue response. Thank you for joining us.